Welcome to AMiNDR, a podcast where we summarize the latest publications on neurodegenerative disease research so that you can stay up to date with the newest findings. Every month, our team of scientists will sort and organize the titles into themes and present shortened versions of the abstracts. We'll make sure to mention the title, the journal, the first author, and the last author for each publication. Whether you're in the lab, on the bus, or cooking your meal, we hope you find this podcast helpful.
Hi, and welcome to another episode on AMiNDR. My name is Marcia, and I will be your host for this month's section on treatment and testing. All the papers in today's episode involve animal model studies, and there are 21 of them. So without any further ado, let's get started. First up, here are two papers where the authors target metabolism or lipids in their treatment approach. The title of the first paper is Integrated Metabolomic and Lipidomic Analysis Reveals the Neuroprotective Mechanisms of Bush-Tiancio Formula in an amyloid-beta 142-induced rat model of Alzheimer's disease. The first author is Yi, and the last author is Peng, and this paper was published in the journal Oxidative Medicine and Cellular Longevity. In this study, the researchers evaluate the effects of Bush-Tiancio Formula, or BST-SF, on cognitive performance and on metabolomic and lipidomic profiles of cerebral cortices in AD rats. They identify 47 metabolites and 30 lipids which they associate with the following: sphingolipid metabolism, glycerophospholipid metabolism, and linoleic acid metabolism. These are significantly altered in the cerebral cortices of rats. Their analysis also revealed 34 differential endogenous metabolites and 21 lipids as potential therapeutic targets of BST-SF against AD. They conclude that lipid metabolism is highly dysregulated in AD rat cerebral cortices and that BST-SF may restore metabolic balance through its neuroprotective action. The next paper is titled Studies on the Neuromodulatory Effects of Ginkgo biloba on Alterations in Lipid Composition and Membrane Integrity of Rat Brain Following Aluminum Neurotoxicity. This paper was published in the journal Neurochemical Research by first author Verma and last author Nehru. Aluminum is a potent neurotoxin that has been linked to Alzheimer's disease-like pathology. It can affect oligomerization and can cause conformational changes of proteins by binding to the lipid bilayer in cells. The authors of this paper expose rats to aluminum lactate in the hippocampal and cortical regions. They then evaluate the influence of Ginkgo biloba on lipid profile alterations using FTIR spectroscopy. They find a significant decrease in polyunsaturated fatty acids, membrane fluidity, and levels of cholesterol, sphingolipids, and phospholipids in the aluminum-treated groups. In contrast, they observed that aluminum treatment increased the fluorescence polarization, anisotropy, and order parameter, but it was normalized following Ginkgo biloba supplementation. From their results, they conclude that Ginkgo biloba pretreatment has a beneficial influence on the adverse effects caused by aluminum exposure. Next up, we have a paper on the gut microbiome. The title of the paper is Yeast beta-glucan alleviates cognitive deficit by regulating gut microbiota and metabolites in amyloid-beta 142-induced AD-like mice. The first and last authors are Zhu and Liu respectively, and this paper was published in the journal International Journal of Biological Macromolecules. Here, the researchers aim to investigate the effects of yeast beta-glucan in Alzheimer's disease. They do so by treating amyloid-beta 142-induced AD mice with small and macromolecular yeast beta-glucans. Using 16S rRNA sequencing and GC-MS respectively, they find that yeast beta-glucans cause intestinal flora alterations and produce short-chain fatty acids. They especially observe compositional changes in some beneficial and inflammation-related bacteria. They perform correlation analysis and see that the microbiome is strongly associated with AD-related pathologies and cognition. Finally, they also find that both small and macromolecular yeast beta-glucans improve neuroinflammation and brain insulin resistance, which goes to show that yeast beta-glucans as a dietary supplement may prevent early-stage AD pathology. Now, we're moving to a section where the target is autophagy. Autophagy is a process by which components within cells are broken down and recycled. In the brain, autophagy can protect cells against accumulation of proteins that cause neurodegeneration. Within this section, we have two papers. First up is a paper titled Ishan Huaxu decoction induces autophagy to promote the clearance of amyloid-beta 142 in SAMP8 mice, mechanism research of a traditional Chinese formula against Alzheimer's disease. This paper was published in the journal CNS and Neurological Disorders, Drug Targets by first author Wang and last author Seo. The objective of this study was to investigate the possible mechanisms of Yishan-Huaxu decoction, which I'll refer to as YHD for the rest of this summary, against SAMP8AD mouse models. The investigators believe that YHD could induce autophagy initiation, promote amyloid beta clearance, and reduce neuronal loss in AD. From their results of their experiments, they make a number of key observations about the YHD-treated mice. Here are some of their findings. Using the Morris-Water maze test, they find that the escape latency time of YHD group is significantly shorter than the control group on the 4th and 5th days. Using Nissl staining, they find that number of neurons in YHD group increases significantly, and using Western blotting, they also see that the YHD could promote the clearance of amyloid beta 142 protein, improve the expression of Beclin-1 and PCL2 proteins, and reduce mTOR and P62 protein levels. Overall, they conclude that YHD could regulate autophagy, promote amyloid beta clearance, and improve memory impairment in SAMP8 mice. The next paper published in the journal Cell Death and Disease is titled Intraperitoneal Injection of Interferon Gamma Restores Microglial Autophagy, Promotes Amyloid Beta Clearance, and Improves Cognition in APP-PS1 mice. The first and last authors are He and Xiao. Here, the authors are interested in whether cytokine interferon gamma signaling is involved in amyloid beta degradation in AD by autophagy. From experiments conducted in APP-PS1 mice, they find that interferon gamma administration significantly increases LC3 2:1 ratio and autophagic flux, decreases P62 levels and amyloid beta plaque load in the mice cortex and hippocampus, and rescues their cognitive deficits. Using intravital 2-photon microscopy and fluorescent staining, they also find that microglia interact with exogenous interferon gamma and amyloid beta, and surround amyloid beta in APP-PS1, CX3C1 GFP mice. Based on these results, they suggest that peripheral injection of interferon gamma might restore microglial autophagy and induce amyloid beta phagocytosis in AD. In this coming section, we have 3 papers targeting mitochondria as an approach in treatment. The first one is titled Protective Effects of Co-Administration of Zinc and Selenium Against Streptozotocin-Induced Alzheimer's Disease, Behavioral Mitochondrial Oxidative Stress, and GPR39 Expression Alterations in Rats. It was published in the journal Neurotoxicity Research with first author Farbood and last author Nawabi. Trace metals such as selenium and zinc have key roles in AD pathogenesis. The researchers of this study are interested in evaluating the effects of co-treatment of zinc and selenium against streptozotocin-induced AD. They subject male Wistar rats to the combination treatment, followed by behavioral tests, mitochondrial oxidative stress, and GPR39 gene expression evaluations. They find that co-treatment with zinc and selenium improves cognitive performance, mitochondrial dysfunction, and oxidative stress caused by streptozotocin-induced AD, but they see no significant changes in GPR39 expression. Based on these findings, they conclude that improving mitochondrial function could prevent AD initiation and progression. The title of the next paper is Beta-acerone improves learning and memory in amyloid beta 142-induced Alzheimer's disease rats by regulating PINK1 Parkin-mediated mitophagy. It was written by first author Han and last author Fan and was published in the journal Metabolic Brain Disease. First, the researchers induced AD model rats by injecting them with amyloid beta 142, after which they subject them to beta-acerone treatment. As previously shown by this group, beta-acerone reduces amyloid beta expression in AD mouse models. In this study, they used the water maze test to evaluate spatial learning and memory and find that beta-acerone treated rats showed improved learning and memory compared to the model group. They also measure hippocampal BCL2 levels and expression of amyloid-beta 142, Beclin-1, P62, PINK-K1, and Parkin proteins through Western blotting and immunohistochemistry. They find that beta-acerone decreases amyloid-beta 142, BCL2, P62 levels, but increases Beclin-1, PINK-1, and Parkin levels. These results suggest that beta-acerone, by promoting PINK1 Parkin-mediated mitophagy, is a potent treatment for AD. The title of the final paper within this section is Pharmacological Inhibition of Soluble Epoxide Hydrolase as a New Therapy for Alzheimer's Disease. It was written by first author Grin and Ferre and last author Galdiano, and this paper was published in the journal Neurotherapeutics. Here, the investigators examined the efficacy of the enzyme soluble epoxide hydrolase, or SEH for short, as a treatment method for AD. They used models of age-related cognitive decline in AD and ACH inhibitors as chemical probes to test cognitive impairment and other hallmark AD pathologies. They see that ACH is upregulated in AD patient brains and that its inhibition reduces cognitive impairment, neuroinflammation, tau hypophosphorylation, and amyloid plaque formation. Based on these results, they conclude that targeting ACH is a potential strategy for AD treatment. That wraps up our targeting mitochondria section. I'll be back in just a moment. Up next, we have 5 papers targeting oxidative stress. The first paper is titled Tocotrienols Influence Body Weight Gain and Brain Protein Expression in Long-Term High-Fat Diet-Treated Mice. It was published in the journal International Journal of Molecular Sciences by first author Cato and last author Fukui. Researchers of this paper are interested in how obesity and oxidative stress induce cognitive dysfunction, which is a characteristic symptom of Alzheimer's and Parkinson's diseases. They are especially interested in tocotrienols, a group of vitamin E, which have strong neuroprotective and anti-obesity effects. In their experiments, they feed mice a high-fat diet from 9 to 14 months of age and assess the effects of tocotrienol treatment on body weight, brain oxidation levels, and cognitive function. They find that tocotrienols prevent body weight gain and also reduce oxidation levels in the brain of high-fat diet-treated mice. These results indicate that tocotrienols have anti-obesity effects and inhibit obesity-induced brain oxidation. The title of the second paper, published in the journal Drug and Chemical Toxicology, is Characterization and Neuroprotective Properties of Alkaloid Extract of Vernonia amygdalina deline in Experimental Models of Alzheimer's Disease. The first author is Obo and the last author is Ogunyusui. In this study, the authors aim to examine the neuroprotective properties of bitter leaf alkaloid-rich extract, which I'll refer to as the extract for the course of this summary, in transgenic fruit fly model and in scopolamine-induced amnesia rats. They treat fruit flies expressing human APP and base1 genes and amnesia rats with the extract to determine survival rate and key neuronal enzyme activity. From the results of their experiments, they see improved survival rates and activities of base1, acetylthiocholine, glutathione S-transferase, and catalase in the extract-treated fruit flies. In the extract-treated amnesia rats, on the other hand, they find that antioxidant status and activities of acetylthiocholine, butyrylcholine, monoamine oxidase, and NTPases is improved. The authors thus conclude that bitter leaf alkaloid-rich extract may have a curative role in neurological diseases like AD. Next up in this category is a paper titled MOF Encapsulated Nanoenzyme Enhanced siRNA Combo Control Neural Stem Cell Differentiation and Ameliorate Cognitive Impairments in Alzheimer's Disease Model. It was written by first author U and last author Q and published in the journal Biomaterials. Neural stem cell transplantation is used as treatment for many neurodegenerative diseases. However, the transplanted neurons are severely damaged by oxidative stress, hindering neurogenesis. In this paper, the authors tackled this issue by using nanoenzymes to promote nerve regeneration. They do so by integrating antioxidative nanoenzymes into metal organic frameworks which are loaded with small interfering RNA and retinoic acid. The metal organic framework promotes neuron directional differentiation, while the integrated nanoenzymes eliminate ROS and prevent oxidative damage to newborn neurons, thereby increasing survival rate and neuronal outgrowth. They test the efficacy of these nanoparticles in triple transgenic AD mouse models and find that it promotes neurogenesis and improves cognitive function. The next paper, published in the journal Plant Foods for Human Nutrition, is titled Neural Protective Effects of Millet and Millet Polyphenols on High-Fat Diet-Induced Oxidative Stress in the Brain. The first author is Lee and the last author is Liu. In this study, the researchers explored the antioxidant and neuroprotective functions of millet on oxidative stress induced by a high-fat diet in rat brains. They find that millet intake reduces oxidative stress in the hippocampus and downregulates the expression of the AD-related genes such as APP, tau, and gamma secretase. They find that millet polyphenols also significantly prevented peroxide-induced cell death of SHSY5Y cells, decreased oxidative stress levels, and downregulated expression levels of pro-inflammatory factors and AD-related genes. Based on these results, they conclude that millet and millet polyphenols could exert neuroprotective effects under high-fat diet-induced oxidative stress. The last paper within this category is titled Bajetian-1 Attenuates D-galactose-induced memory impairment and bone loss through suppression of oxidative stress in aging rat model. It was written by first author Zhu and last author Zin and was published in the journal Ethnopharmacology. Bajetian-1 or BJTW is a Chinese formulation commonly used for the treatment of osteoporosis and AD. In this study, the authors examined the role of BJTW in neuroprotection and prevention of bone loss induced by D-galactose. For their mouse model, they inject male Wistar rats with D-galactose and then treat them with BJTW for 4 months. Using the Morris water maze test, they find that BJTW treatment significantly reduces the latency time for finding the target platform in the directional swimming test and increases time spent swimming in the target quadrant with the probe test. Using microcomputed tomography, they scan rat femurs and find that BJTW treatment reduces D-galactose-induced bone loss. They suggest that this happens by regulating alkaline phosphatase, osteocalcin, osteoprotegerin, and receptor activator or NF-kappa B ligand levels. They also observe that BJTW treatment increases serum catalase and glutathione peroxide levels, reduces hippocampal malonyl aldehyde, and upregulates expression of Fockhead O1, which upregulates superoxide dismutase 2 in the femur. From this, they conclude that BJTW is a potential treatment for AD and osteoporosis. In the next category, we talk about papers targeting inflammatory processes and glial cells. We have 8 papers here, so let's get right into it. The title of the first paper is Xylostazol Restores Autophagy Flux in Bifidobacterium A1 treated cultured cortical astrocytes through lysosomal reacidification. Roles of pKa, zinc, and metallothionein 3. It was published in the journal Science Reports by first author Kim and last author Ko. Xylostazol, a phosphodiesterase 3 inhibitor, reduces amyloid-beta levels in AD mouse models. The researchers of this study are interested to see if xylostazol also reduces lysosomal dysfunction. Here, they observe that astrocytes treated with bifidobacterium A1 shows lysosomal alkalization, which is reversed on xylostazol addition. This also increases free zinc levels, but when a zinc chelator is added, it nullifies lysosomal reacidification. In contrast, in metallothionein 3 null astrocytes, they find that xylostazol addition does not reverse bifidobacterium A1 mediated lysosomal alkalization or increase free zinc levels. This indicates that zinc increase was primarily related to metallothionein 3. The authors make similar observations in FITC amyloid-beta treated and GFP MHTT-Q74 expressing astrocytes. Check out the original paper for more details. Next up is a paper titled Comparison of the protective effects of ginsenosides RB1 and RG1 on improving cognitive deficits in SAMP8 mice based on anti-neuroinflammation mechanism. It was written by first and last authors Yang and Wang respectively, and this paper was published in the journal Frontiers in Pharmacology. The objective of this study is to examine the effects of ginsenosides RB1 and RG1 on improving cognitive deficits in SAMP8 mice. The authors give the mice ginsenosides and donepezil, followed by Morris-Water maze tests, object recognition tests, and passive avoidance experiments. They also perform HE staining, immunohistochemistry, and Western blot experiments to study the possible mechanisms of RB1 and RG1. Based on their experiments, they make a number of important observations. Here are some of their findings. They see that RB1 and RG1 treatment improves the discrimination index of SAMP8 mice in the object recognition test. It also reduces the number of errors in the passive avoidance task. Using Western blotting, they find that over RG1, RB1 decreases protein levels of ASC, caspase-1, and amyloid-beta in mice hippocampus. But RG1 is more effective than RB1 in decreasing iNOS protein levels. Overall, they conclude that both RB1 and RG1, through different mechanisms, may have a role in improving cognitive impairment in AD mice. The title of the next paper is Fasudil inhibits the activation of microglia and astrocytes of transgenic Alzheimer's disease mice via the downregulation of TLR4, MyD88, NF-kappa B pathway. It was published in the journal Neuroimmunology by first author Gua and last author Ma. Activated microglia can transform astrocytes to a neurotoxic A1 phenotype, which is associated with neuronal damage as seen in AD. In this study, the researchers evaluate the ability of Fasudil treatment in overcoming this neurotoxic phenotype using AD mouse models and in vitro studies. They see that in mice with microglial activation and neurotoxic A1 astrocyte phenotype, treatment with Fasudil improves cognitive impairment, inhibits microglial activation, and shifts astrocytes from A1 to neuroprotective A2 phenotype. They find that these effects of Fasudil are through the inhibition of TLR4, MyD88, and NF-kappa B, which are key mediators of inflammation. Their in vitro experiments also show similar results, making Fasudil a potential candidate for AD treatment. The next one is titled Capturing Amyloid-beta Oligomers by Stirring with the Microscale Ion Oxide Stir Bars into Magnetic Plaques to Reduce Cytotoxicity Towards Neuronal Cells. It was written by first and last authors Tsai and Xu respectively and was published in the journal Nanomaterials. In this paper, the authors built on previous findings, pointing the finger at amyloid-beta oligomers as a major culprit in neuronal death and inflammation. The researchers developed a novel method of AD therapy using a silica-coated iron oxide stir bar. On magnetic stirring, it captures soluble amyloid-beta oligomers and converts them into magnetic plaques. These magnetic plaques are then cleared by activated phagocytic microglia, thereby reducing neurotoxicity towards neuronal cells. The researchers also find that neurons treated with soluble amyloid-beta oligomers under magnetic stirring show comparable neuron-specific protein expression to those in absence of soluble amyloid-beta oligomers. In addition, they also see that the phagocytic uptake of magnetic plaques by microglia is greater than that of soluble amyloid-beta oligomers. As a result, the inflammation induced by pro-inflammatory cytokines is lessened. The next paper is titled Anti-Neuroinflammatory Effect of Short-Chain Fatty Acid Acetate Against Alzheimer's Disease via Upregulating GPR41 and Inhibiting ERK, JNK, and NF-kappa B. It was published in the Journal of Agricultural and Food Chemistry by first author Liu and last author Sun. The aim of this study is to examine the effects of neuroprotective short-chain fatty acid acetate on AD. For this, the investigators administer acetate to a male APP-PS1 transgenic mouse for 4 weeks. They test cognitive function, microglia activation, levels of CD11B, COX2, and G protein coupled receptor 41, and phosphorylation of ERK, JNK, and NF-kappa B, P65. Based on their results, they observed that acetate reduces cognitive impairment and decreases CD11B levels. They also find that in amyloid-beta stimulated BV2 microglia, acetate inhibits phosphorylation of NF-kappa B P65, ERK, and JNK, decreases COX-2 and interleukin-1 beta levels, and decreases GPR41. Thus, through its anti-neuroinflammatory effects, acetate might be a candidate for AD treatment. The next paper is titled Low-Dose Ionizing Radiation Modulates Microglia Phenotypes in the Models of Alzheimer's Disease. It was published in the International Journal of Molecular Sciences. The first and last authors are Kim and Chung respectively. During AD progression, microglia can be polarized from anti-inflammatory M2 to pro-inflammatory M1 phenotype. This can be reversed by the activation of triggering receptor expressed on myeloid cells 2, or TREM2, which reduces amyloid-beta deposition and memory loss in AD. In this paper, the authors examined low-dose ionizing radiation, or LDIR for short, as a treatment method for AD and its role in M1-M2 polarization. They confirmed that LDIR treatment in mouse models for AD reduces amyloid-beta deposition and improves cognitive deficits using thioflavin S staining and Morris-Water-Mace test respectively. They next test the effects of LDIR on LPS-induced M1-M2 cytokines in BV2 microglial cells. They find that in the LPS and LDIR-treated BV2 cells, the M2 phenotypic marker CD206 is increased compared with LPS and sham-treated BV2 cells. In addition, they study the effect of LDIR on M2 polarization and find increased expression of TREM2 in LPS-induced BV2 cells. Overall, they conclude that LDIR has beneficial effects in AD-related pathologies. The next paper published in the journal Aging is titled Salvionilic acid B improves cognitive impairment by inhibiting neuroinflammation and decreasing amyloid-beta level in Porphyromonas gingivalis infected mice. It was written by first and last authors Liu and Sun. Porphyromonas gingivalis, or P. gingivalis, the pathogen of chronic periodontitis, causes amyloid-beta accumulation, a characteristic AD pathology. To protect against P. gingivalis-induced cognitive impairment, the authors of this paper investigate the neuroprotective role of salvionolic acid B, or SALB, using a P. gingivalis infected mouse model. They find that SALB treatment shortens the escape latency and improves percentage of spontaneous alternation in the mice. They also see that the SALB reduces levels of amyloid-beta, ROS, malone dialdehyde, interleukin-1 beta, interleukin-6, and RAGE. In addition, they observe that SALB increases levels of antioxidative enzymes, mRNA levels of BDNF and NGF, and the protein expressions of ADAM10. Overall, they conclude that SALB could successfully inhibit neuroinflammation and decrease amyloid-beta levels in P. gingivalis infected mice. If you want to hear more about P. gingivalis in AD, it was mentioned in one of our papers covered in our episode on neuroinflammation and glial cells, hosted by Sarah, as well as another paper in our episode on tau pathology hosted by Nyla. We've reached the final paper within this section. Its title is A Natural Xanthone Alpha-Mangostin Inhibits LPS-Induced Microglial Inflammatory Responses and Memory Impairment by Blocking the TAK1 NF-kappa-B signaling pathway. It was written by first author Guan and last author Li and was published in the journal Molecular Nutrition and Food Research. In this paper, the researchers explored the activity of alpha-mangostin, a polyphenolic xanthone obtained from mangosteen, on LPS-induced microglial activation and memory impairment. They use enzyme-linked immunosorbent assay, or ELISA, or Western blotting to observe cytokine production, NF-kappa-B, and the activation of transforming growth factor activated kinase 1. They use scratch wound healing assays and phagocytosis of fluorescent latex beads to study microglial migration and phagocytosis respectively. They also evaluate learning and memory abilities of mice using the Morris-Water-Mace test. From their results, they find that alpha-mangostin reduces LPS-induced pro-inflammatory cytokine production, inducible nitric oxide synthase or INOS expression, migration, and phagocytosis in microglia, and that it also rescues neuronal dendritic damage. They also find that alpha-mangostin suppresses LPS-induced toll-like receptor 4 expression and TAC1 and NF-kappa B activation. Owing to its anti-neuroinflammatory, neuroprotective, and memory-improving effects, the authors conclude that alpha-mangostin is a potential AD treatment strategy. This brings us to the end of this episode, where we focused on animal model studies. If you're interested in papers on in vitro studies, be sure to check out our episode on novel drug development hosted by Ellen coming out soon. And as a reminder, if you would like to receive our bibliographies, please send us an email or fill out a form. You can also find more details about this on our episode notes. Thanks for tuning in. Hope you enjoyed it.
That's it for this episode. A huge thank you to the team that is working on sorting, summarizing, and scripting these abstracts, as well as the operations behind AMiNDR. The music is from Journey of a Neurotransmitter by Anusha Kamesh, musician and fellow scientist, and now member of the AMiNDR team. You can find the original piece and her other music on SoundCloud under Anusha Kamesh or on her YouTube channel, AK Music. Interested in joining the team? Give us a shout! We can always use help with content development, podcast editing, advertising, and you can be part of a new and exciting venture. Reach us by email at aminderpodcast@gmail.com or follow us on Twitter. Oh, we're also on Facebook now. Don't forget to subscribe to our mailing list if you want access to the bibliography for each of our episodes. The references come with timestamps. Timestamps. So you can more easily locate the paper that caught your interest. Check our notes below for details on how to sign up. And very close to this, you'll also find a link to our feedback survey. Because, your feedback matters to us. So please, pretty please, let us know how we can make this podcast a better tool for you. And last but not least, thank you for tuning in with us. And on this note, we hope you found our podcast useful and accessible. Until next time.